All right, now moving to a little bit more laboratory based. Next, we have Shirley Tsunoda, who's a PharmD, Professor of Clinical Pharmacy, Associate Dean for Pharmacy Education in the Skaggs School of Pharmacy and Pharmaceutical Sciences at UC San Diego. And she's going to talk on the intersection of maternal therapeutics and the human milk metabolome.
Thanks so much, Tina. Good morning. I'm happy to be here to talk about a study that we just completed. I'm actually also one of the UCSD CET PIs. And so this was a study. Advance. Sorry. I'm pressing all the buttons. Oh, the biggest one. So this was a study conducted by 3 talented postdocs in our group, and it was funded by a support pool grant. So Sydney Thomas was our previous postdoc scholar. She's moved on now to industry. Simone Renwick, another postdoc in the Bode lab, and then Simone Zuffa, who's here in the audience, who did the microbiome work on this study. So I want to we've been talking a lot about lactation this morning, and, in recognition of the amazing work that's been done to look at the safety of drugs as they go through the mother and into the baby. And, LactMed, for example, is an amazing resource. I want us to think a little bit about looking a little deeper into what are drugs doing to actually the breast milk components, and does that actually impact the infant? And so we conducted a study, kind of, looking at this triad, right? So we have the mother, we have the breast milk, and we have the baby. And this is a dynamic triad where changes that happen to the mother can impact the breast milk, which can then impact the baby. These can be environmental disruptions, such as drugs, right, that the mother takes. And we are learning more and more about the human milk components, as well as the microbiome in the breast milk and what those interactions look like. And does that actually impact infant health? And we've used emerging computational methods to sort of look at this issue. So focusing in on HMOs, or human milk oligosaccharides. These are indigestible sugars that form the 3rd largest solid component of breast milk. So after lactose and lipids, HMOs comprise the 3rd largest component. There are more than 200 species of HMOs known, and they are basically, a complex molecule that is indigestible by the infant, but is used by the gut microbes in the breast milk, often as food. And so, as many of you know, HMOs now have a lot of known biological functions. So they are considered prebiotics, meaning they feed some of the bacteria in the developing infant. And for example, the bifido genera of bacteria is a very important initial species of bacteria that seeds the infant microbiome. And some of the HMOs feed this important bacteria to allow it to grow and develop into the infant's microbiome. We also know that HMOs are antibacterial, so they help to ward off pathogens that may erupt as that microbiome is developing. We also know that sialic acid HMOs are important for infant brain and development. We know that HMOs are important in the development of the immune system and also in the development of allergies and asthma. And then finally, I'm sure you're all aware that it's breastfeeding and the components in breast milk are important in preventing NEC. And so, with breastfeeding, increased in breastfeeding causes a decreased incidence of NEC. And there are components of HMOs such as DSLNH, where higher levels of DSLNH portend lower risk for NEC. And this is just, showing you that there are some mechanisms that are thought to be evolved that allow HMOs to play important roles as that infant gut microbiome is developing. So HMOs both support and inhibit growth of microbes depending on the microbe. And so this is how, again, some microbes are selected to become commensals in the microbiome and others are inhibited so that they don't become pathogens. Additionally, HMOs can serve as pathogen decoy receptors, so preventing certain potential pathogens from taking up residence. And then finally, HMOs have been shown to play roles in decreasing intestinal membrane integrity. So, but we lack a lot of understanding, right, about these complex interactions between HMOs, microbes, and the human milk environment, and does this impact infant health? So we wanted to ask the question, how do HMOs interact with microbes in human milk? And do these interactions have important clinical implications? So we conducted a multi-omic analysis across 3 continents. So we utilized samples from Tina's Human Milk Biorepository in San Diego, samples from Spain, and also samples from Australia. And we conducted a multi-omic analysis. So we conducted 16S rRNA sequencing of the microbiome. We deployed our untargeted metabolomics pipeline to look at the metabolome. And then we utilized HMO analysis to quantitate various HMOs in the breast milk. And what we found was that of 19 HMOs that we were able to analyze, these correlated across the different countries, so Australia, Spain, and the US. And the Shannon Diversity Index, which is a measure of alpha diversity, showed some correlations that were sort of center specific, but also centered around secretors versus non-secretors. And so we actually ended up grouping the data and looking only at the secretor data. The non-secretor data actually didn't show any correlations with any of the environmental or medical conditions that we looked at. And we also noticed that there were 4 HMOs, 6SL, DFLNH, DSLNH, and LSTC that correlated in the same manner across all the geographic sites. So we grouped these into 6 DDL, and I'll be sort of talking about this grouping of 4 HMOs as 6 DDL as we continue through looking at the data. So we looked at various factors, both, parent factors, breastfeeding factors, and infant factors to see how these HMOs associated with these different factors. And so we found some interesting observations with the 6-DDL that 6-DDL did not associate with parent age, but it did associate with parent BMI. And also interestingly, we found that the rate of exclusive breastfeeding was lower in patients with high amounts of 6-DDL, but breastfeeding frequency was also higher in that population. And we sort of hypothesized that this is perhaps because sometimes, mothers who are having difficulty breastfeeding will try to breastfeed frequently, and that this might cause them to give up breastfeeding altogether. So that was just sort of an observation there. And then we also found that 6-DDL decreased with child age, and that the rate of decay was actually higher in preterm infants. And just to note, we only had preterm infants in infant data in the US cohort. And then we wanted to look at how these 6-DDL concentrations associated with microbes in the breast milk. And as you can see from this plot, there were various different, first of all, from a phylum level, all of the geographic regions had the same changes in phylum, the same distribution of phyla, which was, sort of, I guess, a good result that, there's not a lot of large changes across the geography, but when we look deeper at the genus level, there were differences across the different regions. But two genus species in particular, Rothia and Vianella, tracked both across geography and also in the negative direction with 6-DDL. And not only that, but Rothia and Vianella were also strongly associated with each other across all the geography, across all three continents. And Rothia and Vianella were less abundant in milk with high 6-DDL. And then interestingly, milk with high concentrations of 6-DDL were also twice as likely to not have any Rothia or Vianella. And then how does this impact, child or infant health? So we actually had a lot of metadata associated with these samples, and we were able to see scores on the ITSEA questionnaire that parents fill out on their children. And we showed that, and we saw an association with milk that had high 6-DL. Those infants actually scored higher on overactivity and aggression in this ITSEA questionnaire. And then interestingly, bringing in Rothia and Vianella, we did a random forest model to try to predict, does 6-DDL and Rothia Vianella absence or presence predict preterm birth, C-section, or parental SSRI use? And in this random forest model, we actually showed that, yes, these things tracked with both preterm birth and parental SSRI use. The C-section was a little less robust in terms of the model. And then to look even deeper into this issue, Simone Renwick in the Bode lab took two commercially available HMOs, so the 6SL and the LSTC, which were two of the HMOs that were part of that 6-DDL cohort that we looked at. And she grew them and she tried to look at the growth of Rothia and Vianella species in an in vitro environment. And interestingly, she found that those two HMOs actually inhibited the growth of the Rothia species. They did not impact Vianella. And then furthermore, she took pooled HMOs, and you can kind of see the orange line. The pooled HMOs may have actually kind of recovered some of that Rothia growth, as you can see in those graphs. So in summary, it looks like human milk bacteria and HMOs are co-regulated. That was what our conclusion was from our study, and seem to also be associated with both parent and infant outcomes. These 4 HMOs showed strong negative associations with Rothia and Vianella and breast milk, and high levels of 6-DDL and low levels of Rothia were also associated with various parent and infant outcomes, such as C-sections, preterm births, the use of SSRIs and pain relievers, as well as migraines in parents, and also worse scores on this externalizing subsection of the cognitive assessment, showing more aggression and impulsivity behavior in children. So this is just to sort of illustrate kind of the bigger picture that we can use these multi-omic analyses that incorporate both the metabolome, the microbiome, and milk analytics to provide insight into maternal pediatric physiology and pharmacology. And finally, we can start to unravel some of these relationships between microbes, HMOs, and other metabolites in human milk, which may pave the way for more optimal use of medications in this patient population. And so going beyond sort of thinking about a dose to an infant, but really thinking about, is this impacting the infant developing microbiome, particularly during that very early period of development, when we know that there's data to show that that developing microbiome and how that is seeded can impact the infant and the child and the adult life later on. So I just want to acknowledge all the people that worked on this study. This, is multi-omics, is a team science and requires the expertise of lots of different people. So I just wanted to, and I also want to acknowledge MPRINT support pool funding for the funding for this study. Thank you.
Yeah, great talk. I mean, I think this really builds on some of the things that we talked about last year at the second annual MPRINT meeting, which is studying medications and lactation isn't just about does the drug get to the infant and can have an effect, but how is it affecting human milk? And I think you've demonstrated that very clearly here. One question in terms of SSRIs, which seemed like they had, I think, almost the strongest effect. Were you able to look to see, is it a total class effect? Is there any differentiation between SSRIs? And related to that, do you have any thoughts on the mechanism? I mean, is it related to SERT inhibition or is it associated more with depression within the mom?
Yeah, so Erin, that's a great question. I don't have an answer for you. I will tell you that I have hopefully she's listening on this call. She's a clinical pharmacology fellow at UCSF, who we're hoping will get a support pool grant to actually look at this very question. Because as we've seen in our data, we have also more data with 2,500 breast milk samples from Tina's biorepository, where SSRIs are the most common drug that we see in the breast milk. And that's our question. Is it affecting the metabolome and the microbiome? And how is it doing that? So we're still, again, at early stages at looking at all of these macro molecules. Hi.
Hi, Camille Thabi with the NICHD. Thank you so much for this talk. I'm new to the world of metabolomics, and I'm learning all about that. So this is very, very insightful. I was just curious if your research or any of the related research that you're working on is looking at dietary supplements, and then some of the, the agents that mothers use to improve their milk supply and how that affects, infant health.
Yeah. So thank you for that question. We always talk about untargeted metabolomics. Sometimes it's a little bit of a fishing expedition, but this is the beauty and the power of untargeted metabolomics, is that we can see all kinds of things that patients don't report. And so we've built a drug library to more effectively annotate drugs and drug metabolites in breast milk. We're building a personal care product library as well to look at all the chemicals that we put on our skin and, take in the environment, in addition to environmental chemicals like plasticizers and things like that. There's another collaboration that we're working on, hopefully that will get funded in this go round as well, to look specifically at those forever chemicals that get into the breast milk. And so to your question, we're actually looking at that as we speak, especially particularly in these 2,500 breast milk samples that Tina has already collected. We're doing a huge basically survey and look at all of those chemicals, as many as we can annotate in those breast milk samples. So more to come on that.
Yeah, just one quick follow-up before it escapes my head. In terms of assembling those libraries, it may be worthwhile to talk to Lauren Alexunas and the Transporter Elucidation Network, because they are also putting together various libraries to look at chemical libraries, including prioritized libraries from the Office of Dietary Supplements. So it would be really useful, I think, if there are at least some similarities and overlap between the libraries, because that would facilitate doing some more mechanistic studies in their models and making sure that, we can actually see what's going on in the human as well as those models.
Okay. Thank you, Erin. And just one more point on that too, like your question about supplements. We also have the ability to look at food and, all the different chemicals that are in food, which is a little crazy and will make you a little paranoid about what you eat, because there's all kinds of interesting chemicals in our food. So that's also, we have the ability to look at that as well. Thank you.
Thanks, Shirley.